Polysaccharides contain many monosaccharides joined together through glycosidic bonds to form a large molecule. Polysaccharides, because of their size, are insoluble in water. And you can see the first polysaccharide that I've shown here, and this is amylose. Amylose is one component in starch, and it's found in plants. Now, amylose has glucose units joined end-to-end linearly, and it coils into a helix. This allows amylose to be compact, so many amylose molecules can be stored in a small area in the leaf. This is helpful because you can store many of these molecules. And if you remember that amylose and starch in general is an energy store. When the plant cannot photosynthesize, glucose units from starch are broken off and they are used in respiration. Now, amylose contains only one type of glycosidic bond, and here this is the alpha 1,4 glycosidic bond. Now here we've got two variants of the name. You can say glycosidic or glucosidic. So please check your specification for which one you should use. Now, as we said before, carbon 6, carbon 5, carbon 4, 3, 2, and 1 in this 6-membered ring, this hexose. And here on the next glucose, 6, 5, 4, 3, 2, and 1. So you can see that carbon 1 here in this first alpha glucose is forming a glycosidic bond to carbon 4 here in the next glucose. So it is a 1 to 4 glycosidic bond. Now in amylopectin is also found in starch. It's another component of starch. And amylopectin does not have a linear structure, it has a branched structure. So you can see that each linear molecule, for example, this linear molecule, will have many glucose units joined together through alpha 1 to 4 glucosidic or alpha 1 to 4 glycosidic bonds. And you can see that this is a linear molecule. At a branch point, let's say this branch point here, you will get one linear molecule branching out into two. How is this achieved? Well as alpha 1 to 4 glucosidic or glycosidic bonds, we also have alpha 1 to 6 glycosidic bonds. And you can see there's carbon number 1 here in this hexose, and carbon number 6 here in this hexose form a glycosidic bond in the same way as before through condensation reactions. So amylopectin is less compact than amylose. The advantage of amylopectin is that because it's less compact and it has many branched ends, it's easier to hydrolyze. So amylopectin can be hydrolyzed more rapidly. So finally, we move on to the polymer cellulose. Cellulose consists of multiple fibers. Here you can see two cellulose molecules, one here and the other here, and they're lined one on top of the other. Multiple cellulose polymers lined up like this form microfibrils. Different microfibrils can come together to form macrofibrils. Microfibrils and macrofibrils are held together by hydrogen bonding between each cellulose polymer. Now, as we talked about beta glycosidic bonds before in a previous video, here we can see that cellulose is made up of beta glucose units. Now we have beta glycosidic bonds, and as you can see, they're changing orientation. Now this means that each alternate glucose molecule is in a different orientation. So if I look here, you can see that this molecule here, this molecule here, and this molecule here are inverted. So each alternate glucose molecule here in cellulose is inverted. What this means is that the OH group can be found on both faces of one cellulose polymer, not only just on one face. What this does is that this maximizes the hydrogen bonding between cellulose polymers. So let me show you how this works. So hydrogen bond, for example, is a bond where hydrogen is shared between O or N atoms. So let's just say that we have a covalent bond between O and H. That hydrogen is now going to be shared through hydrogen bonding with another O or another N. And in this case, the hydrogen is going to be shared between two O atoms because there are no nitrogens in glucose. So we can see that hydrogen bonding can occur, for example, here, between that H and that O. Or here, for example. Due to the nature of the strand where each alternate glucose is inverted, you can maximize the number of hydrogen bonds between the polymer strands. And this strengthens the interactions, making a strong, rigid microfibril. And in turn, when microfibrils interact with each other through hydrogen bonding, a strong, rigid macrofibril is formed. This provides strength to the cell wall of the cell. And as we talked about earlier, cellulose is a major component, a structural component of the plant cell wall, which provides support and structure. Now, the other type of hydrogen bond that can form, for example, could be between this oxygen and an OH group, for example, this OH group. But this CH2OH needs to be in close proximity. So you should be able to draw hydrogen bonds between cellulose polymer strands. In this section of the video, we're going to go beyond the A-level specification. Before we talked about amylose and amylopectin, I want to focus on amylopectin and compare it to glycogen. Amylopectin contains linear strands, such as this, which can be up to 30 glucose units long. Glycogen, which is found in animals, so it's a storage molecule, for example, found in the liver and muscle in mammals. It contains a similar structure to amylopectin. It has a branch structure. It contains alpha-1 to 4 glycosidic bonds and alpha-1 to 6 glycosidic bonds, and it contains only alpha glucose. However, in glycogen, the linear strands are only around 8 to 12 glucose units long. This enables glycogen to be hydrolyzed more quickly. In plants, amylopectin starch and amylose are hydrolyzed by 3 enzymes. These are 3 types of amylase. The first amylase I want to talk about is Beta-Amylase. Beta-Amylase can hydrolyze alpha 1 to 4 glycosidic bonds, and it works from the end of the molecule. The second enzyme I want to talk about is called alpha amylase. Now, alpha amylase can hydrolyze bonds from anywhere in a linear chain, and it hydrolyzes alpha 1 to 4 glycosidic bonds. Both alpha and beta amylase produce maltose. So whereas beta amylase and alpha amylase can both hydrolyze only alpha 1,4 glycosidic bonds, another type of amylase called gamma amylase can hydrolyze alpha 1,4 and alpha 1,6 glycosidic bonds. So it can break up the glucose molecules by hydrolyzing glycosidic bonds like this and like this. These are alpha 1 to 4 glycosidic bonds. It can also hydrolyze alpha 1 to 6 glycosidic bonds at branch points, like this. So we're hydrolyzing these types of bonds and also these types of bonds. It is worth noting that the three amylases here, alpha, beta, and gamma, can hydrolyze bonds in amylose and amylopectin. Versions of these enzymes are also found in animals, for example in humans, where they can hydrolyze glycogen. And they operate on the same types of principles by hydrolyzing the same glycosidic bonds. I hope you found this useful. Please check out future videos that will be posted to this site.